everyone out there in the void. Welcome to our webinar entitled Data-Driven AI Innovations in Glaucoma Research. My name is Shane Liddelow, and I'm an assistant professor at NYU Grossman School of Medicine here in New York City. And I'm in the departments of Neuroscience and of Ophthalmology. And I'm also the director of translation at the Institute for Translational Neuroscience and co-director of the Parekh Center for Interdisciplinary Neurology. In my role with the Glaucoma Research Foundation, who's hosting our webinar today, I'm a member of the Scientific Advisory Board for the Catalyst for a Cure 4 team. But for the scientific programming today, joining us is Dr. Karthik Shekhar. Dr. Shekhar is an assistant professor of chemical and biomolecular engineering at UC Berkeley in the Bay Area in California. Dr. Shekhar's research is really the epitome of interdisciplinary. He integrates computational biology, single-cell transcriptomics, which he'll explain to you today, and neuroscience to investigate the organization and the evolution of important cell types for vision in the retina, as well as in other parts of the brain and other tissues throughout the body. Karthik completed his PhD at MIT in Boston and his postdoctoral training at the Harvard Medical School. And he's received numerous prestigious awards, including a McKnight Fellowship in 2023, and he has been very lucky to receive an NIH K99R00 Career Transition Award. And I will say, Karthik being one of the members of the CFC4 team that I get to work with quite regularly as he works with the other team members to understand not only neurodegenerative diseases as a whole, but really honing on glaucoma, that we're really honored to have him today here as one of our speakers. And I'd like to welcome Dr. Karthik Shekhar to give his presentation.
Thank you. Thank you, Shane, for the very kind introduction. Thank you once again to the Glaucoma Research Foundation, and especially Michelle, for making this happen. And thanks to all of you attendees in the void for taking time out of your daily life to come and listen to this talk. Hopefully, I will try my best to make it worth it. Let me start by making a few remarks. Now, it doesn't escape anyone's notice that AI has now become such a ubiquitous part of our lives. It's disrupting almost every sphere of human activity, and it's no surprise that it's going to do the same for medicine. But I want to be a little bit more broader, and what I want to hopefully give you a flavor of today is that data-driven approaches that rely on large-scale data sets can be extremely valuable. So classically, medical research involved framing a hypothesis, and one had to be extremely smart in framing the right hypothesis and pursuing several years of activity, testing that hypothesis, and then deciding at the end whether it was true or not. The advantage with a large-scale data-driven approach is that now we have a much larger toolkit to test a hypothesis or not, but also a discovery engine to completely land up in an unexpected area and find and formulate a new hypothesis that the data tells you. So I hope to give you some examples of these in the context of glaucoma today. And I want to say that this work, as Shane pointed out, is extremely interdisciplinary. There are several people who I'm going to acknowledge and who I may not get the chance to acknowledge, but have contributed to this research. And I also want to really thank the Glaucoma Research Foundation and the Melza M. and Frank Theodore Barr Foundation for supporting this, because some of this is really high risk. But as you will see, it takes a lot of courage on the part of donors, many of whom are in the audience, to support this kind of work, with the hope that one day we can make the lives of many, many patients better. So today's talk is obviously about glaucoma, but I would like to start a little bit more general. Now, brain-related diseases affect more than 3 billion people worldwide. That's about 1 in every 3 of us. Now, these include conditions like stroke, dementia, migraine, epilepsy, autism. And now neurological disorders are the #1 cause of disability around the world, and the second leading to the cause of death. Now together, and this is quite staggering, they account for 440 million disability-adjusted life years, or DALYs. Now, DALYs represent the total number of healthy years lost due to injury or disease in a given year within a population. So this is across the entire world. And so just as a thought experiment, if we were to distribute these DALYs equally among every person in the population, each one of us, including every one of you and me, would lose 20 days of a year completely to disability. So this is a staggering number, and we have to do something about this. Now, what's equally striking to me and personally motivating is how few treatments we have and how rare any real cure is as far as the vast majority of these diseases are concerned. And this gap is what drives our work to better understand the brain and to find solutions that don't yet exist. Let me also say that when we think of medical breakthroughs, it's tempting to imagine that there's a straight line from the acknowledgement of a disease to a cure. But in truth, many of the most important advances began as open-ended questions in basic science. And I would like to make a few illustrations because now more than ever, there is an urgent need to recognize the importance of basic science and the role that it's played in medical breakthroughs for the last century. So for example, scientists in the 1980s began wondering about bacteria and how they develop immunity against viruses. Now, you might imagine that this has no relation to human health, but this is this basic effort that led to the discovery of gene editing techniques that are today quite ubiquitous. You might have heard of CRISPR-Cas9. And this basic discovery over 20 years or so led to Casgevy, a gene therapy that is now used to cure inherited blood disorders like sickle cell disease and thalassemia. Another example is in the 1980s, scientists were studying how blood vessels grow, and they discovered VEGF. It's a protein that tumors hijack to feed themselves. And this work gave rise to drugs like Avastin for cancer and also Lucentis for wet AMD. Immunotherapy, which is now saving several lives in oncology clinics, grew from research actually at UC Berkeley, a few buildings away from where I'm giving this talk, where scientists were wondering about what causes immune cells to become exhausted and what causes them to stop. So basic research that ultimately led to this discovery of immune checkpoints and finally immunotherapeutic drugs like Keytruda and Chimera. Now, the vast majority of these so-called G protein coupled receptors, they were studied initially in flies for their signaling roles in olfaction and hormones, smell. And they turned out to be the targets of many, many modern medications, particularly for psychiatric and pain medication. And finally, there is optogenetics, which started where scientists discovered this odd algae that had a protein that was fluorescent and is now enabling brain research and early trials, the GenSight trials for vision restoration. So these examples show that the path to treating disease often starts far away from the clinic in basic science laboratories. And so hopefully that provides some context of what I'm going to describe today to you in the context of glaucoma. Now, today we will talk about the eye and glaucoma, and as many people on this call undoubtedly know all too well, glaucoma is an optic neuropathy that is the leading cause of irreversible blindness worldwide. On the left, you see how fluid normally drains from the eye, from the ciliary body through the trabecular meshwork. But when this outflow is stopped, which is what happens in both open angle and angle closure glaucoma, the IOP or the intraocular pressure builds up. Now, the images on the right show what happens next. In the healthy eye, the optic nerve looks nice and flat and pink. In glaucoma, it becomes cupped at the optic nerve head and pale as different ganglion cells, the output neurons of the retina, die. And these cross-section cartoons over here show how rising pressure deforms the optic nerve head and damages these vital cells. And the retinal ganglion cells are the only connection from your eye to your brain. And so when these cells die, one loses vision. Now, today's treatments like eye drops, lasers, or surgery, they mainly aim to lower pressure. But IOP is a risk factor. It's not a cause. It's sort of like a collateral that comes along the way. And so they don't necessarily restore vision. They slow or neither do they save cells that are already at risk. And that's the challenge that we now face. How do we neuroprotect these cells? And so retinal ganglion cells, or RGCs, as I will refer to them throughout this talk, are the output neurons of the eye. They collect visual signals from across the retina, and they finally send them down the optic nerve to the brain. Now, the image in the center shows how pressure acts from all sides on the eye walls. It's like trying to squeeze a little balloon from the inside. And on the right, over here, we see how this damage plays out over time. The patient's visual field becomes increasingly constricted, starting with peripheral vision loss and ending finally in tunnel vision or complete blindness if this goes untreated. Now, as I mentioned, current treatments focus on pressure, but we urgently need ways to protect or even regenerate RGCs using either a combination of neuroprotective drugs, gene therapies, or stem cell-based strategies to truly change the course of the disease. This is the open problem that all of us scientists working in vision science face at the moment. Now, with this introduction, I would like to lay out today's agenda. I will describe 3 vignettes of the work that we've been doing, much of which is supported by the Glaucoma Research Foundation, from the very, very basic questions about vision and the eye and the retina to the somewhat translational. Now, everything that we do in medicine rests on a foundation of basic science, on asking fundamental questions about how the retina is organized and how it works. And so first, I will talk about how diverse the cells are in the retina, using a combination of high throughput molecular profiling techniques from transcriptomics and computational models rooted in statistics and machine learning. We're learning that retinal neurons are highly diverse with distinct functions, that each of which is important for some aspect of vision. Now, this diversity of cells matters because some of these cell types are especially more vulnerable in the context of diseases like glaucoma. Now, that brings us to the second question. What makes certain neurons in the retina more vulnerable than others? What makes certain neurons more resilient than others? By studying what puts specific cell types at risk or what makes them resilient, we can think of as a discovery engine to discover therapeutic strategies that protect these cells from dying. Finally, an important question that the field is now confronting, some of which we've contributed to, is how different are human and mouse retinal neurons? Because mouse models are still the preclinical discovery engine for glaucoma, mainly because they are genetically tractable, they are easy to manipulate, and we can experiment on them in a way that is never going to be possible in humans, but also extremely difficult in primate models. So mice continue to be a workhorse for discovery. But it's important to acknowledge, and I'll show you some aspects by which our retinas are very, very different in very important ways. And understanding these differences will help us build better models and ultimately better strategies for treating the human disease, which is what we care about. Now, the retina is often described as an extension of the brain, and for good reason. It's a layered circuit that you can see here that transforms light into electrical signals that the brain can understand. Now, the drawing on the left, which is by the great Spanish neuroanatomist Santiago Ramón y Cajal, shows the retina structure. Photoreceptors on the top sense light, interneurons in the middle process signals, and retinal ganglion cells at the bottom send information to the brain. On the right, you can see a picture of just how diverse these cells are in both shape and in function. And this complexity leads us to a key question. Can we first build a complete atlas or an inventory of the cell types that exist in the retina? And that's the foundation for understanding how the retina works and ultimately what makes or what goes wrong in disease. Now, to understand the diversity of cells in the retina or any tissue, we need tools that can measure thousands of molecules in thousands or even millions of individual cells, because that's the way we grapple or build maps of the cellular diversity. Now, this slide shows how we go from a complex tissue over here to a suspension of single cells in the lab, each of which can be profiled for what's called gene expression or chromatin accessibility. These are just fancy names for molecular features of each individual cell. And suffice to say that from every cell, we can measure up to 10,000 genes and 100,000 chromatin regions. So it's a vast amount of data that we can extract from an individual cell. And now we can do that for hundreds of thousands or even millions of cells in parallel. And so these are vast data sets that have a treasure trove of biological information. And what we want to first understand is how is all this organized? And so on the right, I want to show you just how remarkably fast this entire field of technology development is progressing. It's outpacing even the so-called Moore's law for computer chips. In the past decade, technologies like 10x Chromium and SkySeek and so on have made it possible to profile millions of cells over a few days in a few experiments. I will now describe what these tools actually taught us about the retina. Now, for over a century, neurobiologists and anatomists have classified retinal cells based on their shapes or morphologies, especially ganglion cells, these output cells of the retina, which, as you can see here, show striking morphological diversity. But in just the last few years, single-cell RNA sequencing has allowed us, particularly my group, to catalog these cells by their molecular or gene identities as well. On the right is a map of all the cell types that we've uncovered in the retina of the laboratory mouse, again, used to study glaucoma in many labs. Now, each of these little islands or clusters is a unique type of retinal neuron. You can see how diverse these are in this map. And this now gives us a powerful new framework to study function, development, and also disease vulnerability at the resolution of these individual cell types. We now know each of these molecularly defined types, and we can track them in development and disease. Now that we have an atlas of these retinal ganglion cells, which are the single most important group of neurons for glaucoma, let's now focus a little bit on them and their response to stresses, because that's what happens in any neuropathy. Now, one way we use the retinal atlas that we've built, again, a basic science effort, is to study how different ganglion cell types respond to nerve injury. Now, a common model that's used across many laboratories in the world is what is called an optic nerve crush. It's a mouse model for traumatic nerve injury. It's highlighted here on the left. Although in reality, the optic nerve crush is a model of traumatic injury, it has a few things in common with glaucoma or even optic glioma. Axons are damaged as they pass through the optic nerve head. On the right, you see how ganglion cells look shortly after injury. This is the retina, the uninjured mouse retina, and at 14 or 28 days after injury, most ganglion cells have degenerated. And this clearly seen here in this plot that by 4 weeks, by a week or so, one loses pretty much 80% of all ganglion cells. So pretty much all vision is lost. And this persists, and the cells that die never really return. But I just told you that there are many, many types of ganglion cells. There are about 45 in mice, and not all of them die at the same rate. And so we reasoned that identifying the ones that survive better or die faster may offer clues to resistance, resilience, and also possibly regeneration. Now, what this is saying is that even though all ganglion cells in the eye experience the same injury, they don't all respond the same way. On the left, I'm showing you the same map of 45 distinct ganglion cell types that we've identified. On the right, you can see how they differ in survival after this intervention of optic nerve crush. Some types over here in blue are remarkably resilient, while others in red here are highly susceptible. Now, this raises a key question. What makes one cell type survive while others die? If we can identify the intrinsic molecular or genetic pathways that drive or promote resilience, we may be able to develop a new neuroprotective strategy, a gene therapy that may be applied or delivered extrinsically to all cells to promote their survival, including in a disease like glaucoma. So that was our thinking, that what we had when we began this entire genetic screen. And so in this experiment, by comparing these resilient cell types against these susceptible cell types, we actually identified 65 novel genes that were associated with their survival or degeneration. On the left is the same resilience plot that you saw earlier. On the right are two example genes. One is urocortin or UCN, a gene turned on selectively in resilient cells after injury, and CRHBP in the bottom. It's a gene that is upregulated in vulnerable types. So these kinds of signatures, and I'm just showing you two examples here, could help us uncover intrinsic pathways of neuroprotection because they are part of the gene expression programs that distinguish those cells that survive from those that don't. Now, once we identified genes linked to RGC survival, we asked a simple question: can we actually use them to protect or revive other cells? So on the left, we tested several gene therapies in the optic nerve crush model, overexpressing urocortin or deleting CHRBP, in fact, using CRISPR-Cas9 technologies, significantly improved RGC survival. You can see here, this is the untreated retina where there are very few cells, and any of these treatments significantly increases the number of surviving cells. Now, what's interesting is that these strategies were applied en masse. So even though we're identifying the factors that make a particular cell type survive naturally, by delivering those en masse, we can promote a beautiful neuroprotection that is shared by all neurons. On the right, what I'm also showing you is the regeneration of the optic nerve. Now, both of these treatments, UCN overexpression and CRHBP knockouts, also promoted axon regrowth past the injury site, something that is never seen in the adult CNS. Of course, we need to do much better for these to even be considered for the clinic, but they show that insights from these single cell atlases and these large scale experiments can directly inform therapeutic strategies. And in fact, work in my lab and work in our collaborators' labs actually trying out combinations of these to see if they can, in fact, be better at promoting regeneration. Okay. So far, we focused on RGC types that survive due to changes in their own gene expression. But what about these others highlighted here? They turn out to be highly resilient as well, yet they don't show strong activation of these genes like UCN or CRHBP. So on the right, I'm showing you here that some types, like the ones that I've highlighted in stars, they do express this gene, but there are multiple other types that survive well, but that show no such correlation. Now, this tells us that just intrinsic pathways are not the whole story. So to understand what makes these cells resilient, we need to look elsewhere. And so we hypothesize that perhaps we need to look at extrinsic signals, interactions with other cells in the retinal environment. Now, what do I mean by this? Let me show you a little schematic. So far, we've looked at intrinsic differences, genes inside the cell that predict resilience or vulnerability, but that's only part of the story. Now, some neurons, as shown here on the right, also survive better or die better because of extrinsic factors, signals from neighboring cells or support from the environment. Now, on the right, for example, a neuron receiving neurotrophic support may stay healthy for a very long time, whereas one that loses this kind of support, let's say a signal from a nearby cell, may die very quickly. Even small molecules like dopamine and glutamate can shape cell fate through these beneficial or toxic interactions. Now, to understand these spatial relationships, we needed a different approach. The approaches that I described here involve taking the retina, dissociating it, thereby destroying all spatial context. Whereas here, we realized that we needed to develop a little bit more of a panoramic approach that keeps the retina intact, one that is truly a spatial map where cells are, who they are near, and how they interact can be studied in a very, very facile and high throughput fashion. So this ended up being a 3-year collaboration that was, again, a technology development effort funded by the Glaucoma Research Foundation, where we've now have a technology that I'm going to describe to you. And now we are really excited to apply this to human glaucoma, although all our tests have been on mouse models. Now, again, to understand this resilience in context, we needed to image not just a handful of cells that is often done in histological sections including in the clinical setting. But we wanted to view the entire retinal surface at single cell resolution. This is what I mean by when I say a truly panoramic view. So how does this work? This technology took us about 2 to 3 years to master, and I want to summarize briefly. On the left, we start by dissecting and flattening the retina and tangentially slicing the ganglion cell layer into 5 sections. Now, each of these sections is just about 5 microns thick. That's about 1/20th of the width of a human hair, a single human hair, and it's about 1 centimeter across. So the thinness of these sections makes life a lot difficult in the lab, but we've had to master techniques, and this has happened largely in my collaborator and colleague Shane Liddelow's lab at UCSF. Now then, each section is imaged, and these images are processed and stitched together through a series of AI, statistical, and machine learning algorithms. This is sort of what my lab has pioneered over the last couple of years. And so what you're seeing on the right is a small snippet of this retina, and you can see how rich the information is over here. This is just a 300 micron by 300 micron snippet. It's a zoomed in view of this region of the retina. Every dot here, and there are really tiny dots, is a single molecule of RNA colored by its molecular identity. Now, this approach lets us track the spatial arrangement of all retinal cell types. And as we shall soon see, it also allows us to interrogate how patterns of vulnerability or protection emerge across the tissue. Now, with this approach, we can now capture a panoramic view of the ganglion cell layer, mapping tens of thousands of cells across the retina at about 65% capture efficiency. We can do better, but we're still quite happy with what we've accomplished. The panels above show the distribution of different classes of cells, amacrine cell, ganglion cells, and also non-neuronal cells. The resolution turns out to be so fine that we were even able to trace the retinal vasculature. And this will become very important for what I'm going to describe to you in the next couple of slides. As you can see here, there are these cells arranged in these streaks. And when we look at them histologically, they turn out to be blood vessels, the vasculature. Now, this gives us a detailed anatomical scaffold to interpret cell vulnerability, not just by type, but also by its location and its microenvironment, whether it's close to a blood vessel or an astrocyte or a microglia and so on. Now, the retina turns out to be one of the most metabolically active tissues in the body, and its neurons rely heavily on a dense vascular network for survival. On the left, you see its layered blood supply. There are superficial, intermediate, and deep vascular plexi. These networks deliver oxygen and nutrients to different neurons. On the right are OCT images of angiography images showing how this network is compromised in glaucoma. So for example, what you see here is normal tension glaucoma, angle closure glaucoma, and control. And what's striking is that in glaucoma, there is a striking loss of vascular density in both normal tension and angle closure glaucoma. Now, when vascular support falters, neurons quickly lose metabolic stability and become more vulnerable to injury, even if the intraocular pressure is normal. And so we then used the spatial transcriptomics approach to ask if some types of ganglion cells are more likely to be located near blood vessels. It turned out to our surprise, and this is completely unexpected, and again illustrates the power of these high throughput approaches. There are a large number of these perivascular types that are extremely close to the vasculature, also happen to be the ones that were resilient under optic nerve crush. Now on the left, I am showing you several types over here are the ones that were identified as perivascular, meaning that they were close to capillaries. On the right, you can see this visually. The green labeled cells from this perivascular type are always close to these blood vessels, whereas this is an example of a non-perivascular type. You can see that these bodies are often a few distances far away from these blood vessels. Now, we knew even back then that optic nerve crush is not exactly glaucoma. This therefore raises an important question. Do these perivascular cells actually fare better in glaucoma? And if so, the bigger question is, can we harness that protective niche therapeutically? So the first question we asked is, does being near a blood vessel actually help neurons survive? Now, these data say yes, and to do this, we took the help of Yang Hu, again, somebody who's supported by the Glaucoma Research Foundation, who's developed a beautiful model of glaucoma. This is called the silicone oil model of glaucoma, where it mimics several aspects that are seen in humans. And what I'm showing you here on the top is that when we subject animals to this model, ganglion cells that happen to be close to the blood vessels are much more likely to survive. And this is also seen in both these images and also this frequency plot, where perivascular ganglion cells are enriched very clearly after injury, as you can see by the shift of this distribution from the black to the red. But in the optic nerve crush model, on the other hand, which is again widely used, the effect was much weaker. And so what this taught us is that, again, models matter. We have to rely on animal models to study different aspects of the disease, and it's extremely important for researchers to ensure that as much of the real disease is captured faithfully in these models. Otherwise, we will be using the wrong model to study something that may not be useful in the long run. So the final question that we asked was, are we seeing real effects of perivascular survival or just a survivor bias? So on the left are ganglion cell types that are perivascular even at baseline. After this silicone oil injury, these cells remain enriched in near blood vessels, and their survival also improves. In the middle, we also see these slightly less perivascular types where you see the same trend. But interestingly, on the right, we see that even non-perivascular types, like those labeled with this gene FOXP2, also shift close to vessels after injury show enhanced survival. And so all this is saying that while baseline positioning plays a role, there is also a broader adaptive response, suggesting that these perivascular niches may be protective across multiple ganglion cell types. And although I can't give you the answers right now, it also suggests that we must think about how can we harness this vascular niche in a way that perhaps drugs that normalize the vasculature during glaucoma may actually promote survival. Now, all of this has been, so just to summarize what I've taken you so far, and I'll quickly return to the last part of my talk, which will be hopefully very quick. What I've shown you is that we've built a cell atlas of the retina, especially its ganglion cells. We've identified genetic pathways tied to why some cells survive and others don't. And third, we've also mapped out how spatial position, especially vascular proximity, shapes survival. Of course, all of this that I've described is in mice. The big challenge that I was facing when I started my own lab is how do we translate these insights into human biology and disease? And that's what I want to discuss in the last few minutes of my talk. So here is the problem with mice, in case you didn't know already. The retinas are small, and mice don't have a fovea. The fovea is a specialized central region that is found in primates, including humans, that's responsible for our high acuity vision. And so the human fovea that is shown here is densely packed with cones. It is what we use to read, recognize, and focus on fine details, and mice simply don't have that feature. Now, there is also a very important aspect, is that it's the central vision that enables us to see things at high acuity. And because mice don't have a fovea, they are entirely inapplicable for studying AMD, but they also have serious limitations in studying diseases like glaucoma that are not necessarily macular diseases. And one of the biggest differences arises from the fact that human vision is dominated by this particular group of neurons in the retina called midget ganglion cells. Now, in humans, these midget cells, and they're so called because they have short dendrites, this is in fact directly responsible for our ability to see things at high acuity. In humans, these midget cells make up more than 90% of our visual pathways in the fovea and more than 80% in the periphery. And they are really the backbone of our high acuity vision. By comparison, there are other cell types in the humans that are called parasols and small bistratified cells that are much less abundant. They are important too, but no cell type no single cell type among ganglion cells is as important as the midget cells. Now, the midget cells, as far as we saw in the literature, appear to be unique to primates. It raised the key questions. Do mice or any other animals have functional cells that match these midget cells? If the answer is yes, that's good, because that says that at least some aspects of circuitry is preserved in mice. But if the answer was no, and I was quite surprised that the field didn't have an answer to this question, it raises the question of why are mice being used to study glaucoma in the first place? So to answer this question, again, which is a basic science question, and again, I want to show you here why midget cells matter in glaucoma. They are among the first to show signs of degeneration. In healthy eyes, these cells have fine branched dendrites that collect visual signals that you can see here. But in glaucoma, as you can see below, they rapidly lose dendritic complexity even before dying. And the box plot on the right quantifies this drop in branching. And so if we want to understand and eventually prevent vision loss, we need models that capture the biology of midget cells. This means, first and foremost, identifying their counterparts, seeing if they even exist in species that are routinely used as animal models, and hopefully developing strategies that exploit this in preclinical studies of glaucoma. And so this began, again, an open-ended project in my group to find midget-like cells in other species. We built these retinal atlases across 20 vertebrates, 20 species, from primates to rodents to even lizards and fish. And using methods that we originally developed in mice, we've now profiled a wide range of retinas to search for functional or molecular equivalents of these midget ganglion cells that are so important in humans. And so this work would not have been possible again without a fantastic network of collaborators across Berkeley, Harvard, UCLA, and beyond, and also support from the Glaucoma Research Foundation. The goal is simple, to identify species that naturally encode high acuity circuits and to use them to build more targeted models of glaucoma. That's the vision, and this was also funded by the NIH. So I want to give you the answer because a lot of this is published. Do other species have human, midget, and ganglion-like cells? This map shows that the answer is a resounding yes. Using orthologous gene expression patterns, we found strong molecular matches to these midget cells and parasol cells. What's interesting is that you can see conserved mappings, not just in primates, but in distant species like parrots, ferrets, pigs, and even tree shrews, some of which are used as glaucoma models. Intriguingly, even mice have partial molecular counterparts. It suggests that the elements of these high acuity circuits that have become so dominant in humans are much more ancient than we thought. This is actually good news for studying glaucoma and modeling glaucoma. Now, what's interesting is all of this was based on a machine learning statistical analysis of gene expression patterns, but these cells do not just share gene expression patterns, but also morphological patterns like lamination in the IPL, or even polarity and even functional tuning. And moreover, we found that even the upstream circuits, including bipolar cells, are also conserved. So this suggests overall that despite 90 million years of divergence between humans and mice, that's how long we have to go before the two species share a common ancestor, the circuit logic for visual processing is surprisingly conserved. Now, I want to end by just saying, this is what we learned from all of this. It's all great that we have this conservation, but there is also a very, very important anomaly. Now, rodents, which happen to be the most common models used to study glaucoma, turn out to have only a very, very tiny fraction, just 2 to 4% of their ganglion cells happen to be these midget-like cells. But as we move up to species, including ungulates, carnivores, and others, the fraction of midget cell climbs until it reaches its zenith in primates to about 60 to 80%. Now, this trend isn't random. It reflects the expansion of the visual cortex and the increase in visual acuity and binocular vision. And so the main message is that while rodents remain invaluable, it's also clear that we need to develop alternative genetically tractable models of animals that have a higher fraction of these midget cells, because at least in my view, that's much more promising and likely to translate to a better preclinical prediction towards a clinical trial. So this would, in my view, help find a model that better models human disease than currently the mouse does. So with this, I want to end this talk. Hopefully, I did as good a job as I could to explain 3 vignettes that take us from the basic science of retinal biology to animal models of glaucoma, to even understanding a little bit about how we can take evolutionary inferences by comparing humans and mouse and many other species to develop better models. And I want to end by saying that this work is extremely interdisciplinary. It involves a mixture of genomics, neurobiology, clinical vision science, and also computational algorithms. And it would not have been possible without the possibility of having so many diverse collaborators spread across the world, and also support from both federal agencies and also, more importantly, private agencies like the Glaucoma Research Foundation and the Bar Foundation, for which I'm truly grateful. So with that, I'd like to thank you all for your patience, forbearance, and attention, and I'm happy to hang around and take any questions.
That was absolutely fantastic, Karthik. As always, a super tour de force of all of the really exciting science that's going on that nerds like I get really excited about in your lab, but also touching on, I think, a lot of the really important questions that we're constantly asking about how our research relates directly to clinical studies and to patient outcomes as well. But one of the ones that a few people asked was, you talked about AI and these large data sets in the research and discovery space. Do you think that AI will also be used for drug development, identifying novel compounds, or for optimizing drug delivery, anything like this? What do you think about this space in optimizing basic research to make it more translational for drug delivery and drug development?
The easy answer, it's already happening. It's already happening right where I am. There is a huge push in terms of AI methods and computational chemistry. So if you talk to drug developers, there are a lot of interesting problems. Like, for example, AI has made a huge amount of contribution in understanding protein folding, just as a way to screen for proteins. As people would have heard about AlphaFold, it was a Nobel Prize in chemistry last year. People who design small molecules are also interested in questions of docking and so on. And there are a number of my colleagues who are making huge strides in using diffusion-based generative models, also accelerating what's known as density functional theory calculations for looking at molecular structures and so on. So AI is already making a huge, and 2 days ago, I heard that there's an entire division within NVIDIA now, comprised of about 100 people, that's purely dedicated to accelerating computational chemistry using AI-based tools. So it's happening.
Yeah, I think that was a perfect answer, and I think AlphaFold is the sort of the jewel of this example here. And I would just add to this as well. One of the things that always happens when we're developing new treatments and new drugs is we may have some off-target or some effects that we don't like happening to patients as a result of that drug. And so AI is really driving what we would call the medicinal chemistry side of this, the medchem side of this, which is making multiple variants of each compound, each drug as it is developed, to try and mitigate those off-target or side effects that are occurring. And this is actually just speeding the way that we are able to actually develop these new therapies. They can be made using AI with a knowledge of what that chemistry means. They can be checked for their ability to bind to the target using compound or using tools like AlphaFold, which is really exciting. So I think you're going to hear a lot more in the general press about the application of AI to these sorts of questions. But I think this is a really exciting. I would
just add one more thing. Even though proteins, nominally are made up of just 20 building blocks, and even though most molecules are made up of a finite number of building blocks, if you think about the possibilities that you can get purely based on the number of possible combinations, you will quickly start seeing numbers that are larger than the number of elementary particles in the entire universe. There is this large search space. And one of the reasons people are so excited about AI is to be able to navigate this in a very, very efficient way by almost, some of it is, of course, non-intuitive of why this happens. And understanding why it works the way it works is, again, an active area of research among my computer scientist colleagues. But I would say application-oriented people like me are more than happy to employ them to find answers to questions that were out of reach until a few years ago.
That's fantastic. And maybe what we'll do is I'm going to field one more question on AI, since it's come up in Karthik. You may or may not want to take a stab at this because it's more looking at the clinical side. But, you know, a lot of what we've been talking about, or you've been talking about here today, is, you know, the subjective interpretation of data, right? We need to make sure that we're taking a look at this in an unbiased way. And did you want to make some comments on what you think about the use of AI to manipulate and sort of analyze clinical data, so things like the visual field test, or looking at IOP measurements, or things like this? Did you want to make some comments on that, or would you like to steer clear of that today?
I would actually stay clear of that because it's an area that I don't know much about. But other than, you know, what I would say is any problem that involves truly integrating multi-dimensional, multimodal measurements and discovering patterns, AI will have an application, will have an home. It's completely agnostic to the particular details of, you know, what those measurements are, whether it's clinical measurements, psychophysical measurements, or maybe molecular measurements, as long as one is dealing with high dimensional, high throughput data. Of course, the main constraint that AI models, and especially much more complicated models like really deep neural networks, or even what people call foundation models, the big constraint actually is the data required to train them, right? So you need that. And as long as you have that, there will be a home for AI. But otherwise, if you don't have the data, you have to, it doesn't matter how good a model you have, you just may not be able to train it.
I think that's really fantastic. I think that's a beautiful answer. And the reason I fielded this to Karthik first, even though he's not a clinician, is he really understands this sort of modeling side of things. I'll add one additional point. At NYU Grossman School of Medicine, where I'm based, our clinical imaging core does a lot of use of AI for understanding the small eccentricities of X-ray data, MRI data, CAT scan data, things like this. Again, it is taking the human bias out of the interpretation of these data and looking for very early prognostic things that are happening in those images. So an example here, we're talking about visual field tests. AI algorithms that are properly trained, just as Karthik said, could be used to detect and monitor trends in visual field indices. So you're looking at things like mean deviation, which some of you may have heard of, or pattern standard deviation, essentially how that is changing over time. And ideally, they're going to identify these sort of statistical trends, whether or not it's declining or increasing, and looking at what might be an appropriate intervention for a clinician to take with this additional tool at their disposal. So watch this space. I'm sure there's lots of people that are working on this moving forward. So what I might do, I'm just going to ask, you know, Kate was answering this question offline, but we're going to make a really nice, easy one for you here, Karthik. In your beautiful image of all the different species that you've been working on, you had a squirrel, which I really enjoy talking about, right next to a primate. And so were you suggesting that the squirrel was the second best model here, or you just had a random assortment of animals?
Oh, it's just a random assortment. It's a random assortment of things. It has nothing to do with that. The squirrel was a good model because it happens to be a diurnal rodent.
Mm-hmm.
We forget that often the most common laboratory mice that is used even for vision research is a nocturnal animal. And so squirrel is maybe slightly better than a laboratory mouse, but definitely not as good as a primate.
Yeah, again, that was super perfect. And just for those following at home, one of the reasons that researchers use mice is we have a lot of genetic tools at our disposal, so we can actually be very, very specific in our interactions. We can go on and intervene with individual genes that make individual proteins, like Karthik was talking about, oftentimes in very individual subtypes of cells, like those 45 different types of cells we talked about, and those midget cells. Whereas, when we start making small molecules and drugs, as I mentioned, there can be some off targets. And so one of the reasons a lot of these things start in mice is we have the high fidelity genetic access. Just changing avenues here a little bit, Karthik, we're going to pull on your deep knowledge of engineering approaches and maybe brain-machine interfaces. And just because this question was asked, and I think both of us can talk about this sort of tangentially, what do we think about Neuralink? And what do we think about the possibilities of maybe not Neuralink itself, but brain-machine interfaces as a different intervention here, rather than regenerating retinal ganglion cells, but maybe having an artificial intervention to replace the sort of function of the eye. Do we think that that is on the horizon? I'll let you go first because I'm a very rude host, and then I'm going to make some comments afterwards.
I would say, why don't you go first, because I don't have a good answer, but I have one answer that I can put in from an engineering perspective.
Okay. I think that's fair. So, Pat, I think this is a really good question. And of course, there's a lot of popular press about things like Neuralink brain-machine interfaces. Particularly, this has been really powerful in people who lose function of their forelimbs, maybe after a stroke or a spinal cord injury, or lose the ability to walk, particularly after spinal cord injury, where brain-machine interfaces or spinal machine interfaces and epidural stimulation has been really, really powerful. So there's a couple of ways that one can think about this in the visual system. So one is, would you make a bionic eye? And there's definitely big infrastructural pieces in play across the world, largely based in the US, also based in Australia and in Europe, where they have been trying to make a bionic eye in much the same way that we have a bionic ear that can be used for those people who have lost the ability to have good hearing. There's a few difficulties with this. One is the fidelity of the vision that can be obtained using cameras. Of course, cameras can be very, very high fidelity now, but the power usage can be extremely high as well. So we need to make sure this is of a quality that can be used with a battery pack that is maneuverable around for people. And the second is the interface, exactly like you bring up here with Neuralink, the interface with the higher brain visual systems. And at the moment, we don't have the best brain-machine interface for connecting that really complex visual information with the visual cortical areas of the brain. There is definitely some work that has been done that gives us low fidelity imagery, so the ability to detect objects moving through space, which is incredibly useful for navigating around the world, but it wouldn't give you the high fidelity imagery that you want, or the ability to do this, but it could be requiring a large amount of retraining of that circuitry. So this is a very, very active area of investigation. At the moment, the Neuralink space is not looking particularly at vision, but I'm sure it is something that Neuralink and other commercial systems, as well as academic institutions, are looking at very, very strongly. Now, the other alternative here is not doing a brain-machine interface, but looking at an entire globe replacement. So that would be replacing the entire eye and integrating that back in a biological sense into the circuitry of the higher brain functions. And so there's actually a large group of people actually that are members of GRF earlier CFCs. So Jeffrey Goldberg out of Stanford University is leading a big team, including around 20 institutions across the US, including myself at NYU Grossman School of Medicine, where we are looking at how can we actually do those eye transplants to fully integrate back into the system in a biological sense, to actually replace the vision that may have been lost because the globe has been damaged, or the neural connections that Karthik talked about to the visual system have been damaged, or if the higher brain regions have been damaged as well. So there is a parallel set of investigations going on the brain-machine interface side of things, the engineering side, and on the biological replacement side as well. And I'll hand over to you now as well, Karthik, for your engineering.
I just want to add one thing here. I don't know this space well enough, and I want to comment more as an amateur who looks at it from the periphery than as an expert. Now, one of the very important things when one is designing any kind of brain-machine interface for sensory systems is to look at the information processing speeds. Now, there's one well-known paradox that the eye or any sensory system is able to collect information at a very, very rapid rate, something like 10 to the 9 bits per second in the units of information. But apparently, and this I learned a few months ago, that the processing output of the human brain is actually it's about 8 orders of magnitude slow. It's about 10 bits per second. That's the maximum sort of. So one question is, why is there such a huge gap? And any BMI that is being designed has to contend with the fact that the processing power, because Neuralink is going to be ultimately inserted into the brain somewhere, that the neurons there have a very, very low processing power. And so this gap may exert fundamental physical limits. These are not engineering challenges. These are just physical challenges governed by the laws of the universe, that you may not be able to achieve processing efficiency beyond a certain point. To people interested in more, there's a beautiful paper, perspective paper by Marcus Meister, a famous neuroscientist at Caltech, which is called The Unbearable Slowness of Being in Neuron. It's sort of a play of the novel by Milan Kundera, The Unbearable Lightness of Being. So here he talks about what might be the real challenges or the limits for these brain-machine-like interfaces.
That's really fantastic. Thank you, Karthik. This is really fun because we're coming up from different sides here. So I'm going to ask one question, and this is going to be a what if, and so you can be as spurious as you would like here. And I know this is a very difficult thing to answer, but Sonali had a really important question, I think, we're always thinking about as scientists is, but what do we think about, or what do you think about as the timeline for regenerating cells like the midget RGCs? And maybe we'll start there, and then I'll go to the second part of this question. So what do you think of a timeline for regeneration of a midget RGC?
Oh, by timeline, you mean like how long is it going to take to...
Exactly. How far away do you think that you are in the laboratory setting to be able to do that?
I think we are very, very far away. But again, that's because we have no idea what's in the future. I mean, to people who... I would say, 10 years ago, it would have just been impossible to imagine that AI has the kind of power that we have today. I mean, 10 years ago, if you ask somebody how long would it take to achieve this kind of something, they would have said probably 50 years or 100 years. That's simply because we are not good predictors. We are not soothsayers. And often what happens in science is that progress is exponential. So we just have to keep at it. And it may be that a few things fall in place, and suddenly it could happen maybe even within a 5-year horizon. So right now, I would say the uncertainty is huge. We may be 50 years away, we may be 5 years away, we may be 20 years away, I don't know. But that's just because we are not very good predictors of the future when, so it would be insincere of me to put a number because I have no basis to put that number on.
I think that was a perfect answer, and you actually preemptively answered the second part of the question about how AI is accelerating that, which is really lovely. And I'll just reiterate, scientists, we like not to put timeframes on things because we don't like to promise what we do not know. But just as Karthik said, the exponential speed of which these discoveries are moving forward, because we are always learning so much as a scientific community, interacting with patients to look at what is actually occurring for them and making sure our research efforts are going in the correct direction, as well as now integrating AI into our everyday pipelines at the basic science level and all the way through clinical care, as we talked about earlier today, and also now in the use of developing and optimizing clinical trials and looking at producing personalized medicine programs and really working at making sure that the patient interaction with the clinician and the researchers is vastly improved. And this is really being sped up exponentially, as Karthik said, because of the integration of AI. I want to make just one last
point there, including if you look at the early development of these AI systems, they were not developed with ChatGPT in mind or with in mind the use cases that we are now using them. They were developed because there were scientists, computer scientists interested in basic fundamental questions that might have been dismissed as completely impractical, and indeed they were back then. But they wanted to work at it rigorously, pursue the truth, and end up with something that is absolutely true. And then the history of science tells us that as long as we discover something, however abstract it may seem at present, somehow humanity finds a way to make use of it in the long run to build a practical tool. That's exactly what's happened. And I see research in vision and glaucoma to be no different.
Perfect ending here. And we all know of the most famous example of this, of another recent Nobel Prize, of course, of mRNA and working at mRNA vaccines during the COVID pandemic. So I think with that, since we've run up against time, I just want to thank Dr. Shekhar again for sharing both his time and his expertise. I also want to thank the entire GRF team for their impeccable support in preparing today and for tirelessly working behind the scenes, answering questions as you're going through. And also importantly, I want to thank all of you. I know that we all have a lot of things to get through in our day, and for taking the time to spend with us and providing some really thoughtful comments. I really appreciate this as well. So for all the patients, the family members, friends, and carers on the call, I thank you for taking the time.
Again, I would like to extend my thanks as well. It's really nice of you to spend an hour with us, and it would have been nice to actually have a discussion, open discussion, but some other time.
I reiterate that again. So thank you all for all of you for being here, just as Karthik said, and also for standing and sharing with us. I think GRS has this bold vision of a future free from glaucoma. And as you can see, people like Karthik are really working in these obscure areas, moving forward and trying to gather as much information towards this goal as well. So it's really because of the unwavering support of you that the cure is truly in sight, and enabling the research that Karthik and other members of the GRF CFC teams are able to move forward through. So it's really through your generous donations and your interactions with our researchers that make the work of our dedicated researchers like Karthik possible. This is incredibly critical work, bringing us closer and closer each day to the reality of a world without glaucoma. And as Karthik said, these movements are moving forward at exponential speed now because of the tools at our disposal, including the use of AI. So I'll close here just by saying, if you do have any further questions, please do not hesitate to connect with the incredible GRF staff. Here they all are here. They're absolutely wonderful. They're positioned all across the US, almost every time zone, so we can get your questions at any day of the time of the day or night. They're always here to help. Pass on questions to other researchers if they didn't know, to provide you with additional resources that they have on hand. They are an incredibly passionate, knowledgeable, dedicated team that are here to help you and support you in any way that they can. And with that, thank you very much. Have a lovely end to the week and a really relaxing long weekend as well. Take care.